guys, what is up? It is JLand Bio in the house. Talking a little bit about biology today. Make sure you leave a like and subscribe. Leave a comment down below. Make sure you look for JLand Bio merch coming out here this February. Put in Biology Rocks for 20% off your order. I'm just kidding. This isn't me. Well, I mean, it is me, but I don't talk like this. Hi, guys. Welcome back. We're going to start talking about genetics here in just a couple of moments. If you remember, we talked about in our last several videos the process of mitosis and meiosis. Talked a little bit about independent assortment and how meiosis produces genetically different offspring. Well, that's something that we're really going to focus on in this video today, is how does genetics play a role in who we are as living organisms and the characteristics that we share or don't share with our friends, our family, siblings, and whatnot. So let's get going. So we have quite a few objectives for this particular video, but by the end of it, you should be able to do the following. You should be able to understand how proteins produced in the body result in traits and characteristics that are expressed, explain the key concepts behind genetics, and complete a basic monohybrid cross and analyze for genotypes and phenotypes. So if you've ever heard of the term Punnett Squares or remember the term Punnett Squares, that's something that we're going to take a look at here in this video. So if you have a good idea on it, great. If not, I promise you by the end of this video, you will completely understand how to go through and do that. So let's get going. Oh, look at all the vocab terms we have to do today. Well, there's a lot that's going on here. You know there's a lot of vocab in biology. You should be able to know traits, law of segregation, heterozygous, genotype, Punnett square, genetics, allele, dominant, phenotype, law of independent assortment, gene, homozygous, recessive, and monohybrid. Lots of vocab terms, but let's get going. So how did the concepts of genetics all get started? Well, it started with a man by the name of Gregor Mendel. Gregor Mendel worked with pea plants. He had a garden in the back of the monastery and worked quite a bit with pea plants and determined that inherited traits are not necessarily expressed. So for example, we look at this it's called a pedigree here. But the very top part of the pedigree where it says parent generation, he started with yellow pea plants and bred them with green pea plants. Now, what ended up happening is when he bred his yellow pea plants with green pea plants, they all ended up being yellow. Now, this surprised him a little bit. He expected at least some of them, or as you would predict, maybe 50% of them to be green. If you're breeding one with all yellow and one with all green, you'd get half and half, but all of them ended up yellow. And this perplexed him. So what he ended up doing is he actually ended up breeding two of the F1 generation together. So you see once the yellow and green in the parent generation were bred, there are 4 that are listed there. Now, there were many more pea plants than just 4, but he took 2 of those and bred them together. And with 2 yellows, because yellow is what was expressed, he would expect that all of them would be yellow. But what he got was a ratio of 3-to-1 of yellow to green. Now, this fascinated him as well, so he went and actually did more trials with it and took each of the 4 different types that are down in the F2 generation and bred them together. But what this showed him is that inherited traits are not necessarily expressed, and that determined that traits inherited are independent of one another. So what we can see here is that while he bred yellow and green, all of them in the first offspring had 1 yellow gene and 1 green gene. But only the yellow one was expressed because that's the only one that was seen. All of the offspring were yellow. When he bred the 2 together again, you can take a look down in the F2 generation, you end up with the 3-to-1 ratio of yellow to green. What that told him, again, was that not all traits are necessarily expressed. The green trait that was in the parent generation, that green gene, was passed on. However, it was masked by the yellow one. And the only way that green would ever be expressed is if 2 green genes were inherited. So this gave him quite a few different ideas about some of the stuff that we're going to talk about a little bit later on. But keep in mind that Gregor Mendel worked very, very closely with pea plants, and that helped him determine a couple of the key points that we're going to focus on here in the next couple slides. So what we learned here primarily is that genetics is the study of how traits are inherited in organisms, and different traits are inherited in different ways. You've probably heard of the ability to roll your tongue as an inherited trait. Sorry, probably couldn't see that. A widow's peak, if you take your hair and pull your hair back just a little bit, you might be able to see the hairline kind of goes up a little bit on the sides. That's known as a widow's peak. Attached and detached earlobes. I mean, everything is genetic in some way, shape, or form. Now, traits are characteristics that are inherited. So those are the characteristics we just talked about. Now, organisms inherit 2 copies of each gene, one from each parent. So we talked about in our previous unit that we get half of our chromosomes from mom and half from dad. That means that we get 2 copies of each gene, 1 copy from mom and 1 copy from dad. And these separate during meiosis. That's the law of segregation. So basically what that's saying is that if we look at on the right hand side, if a parent has 2 large Y genes that just represent some particular trait or characteristic, okay, you can inherit one of those 2. You cannot inherit both, but you can only inherit one. If a parent has a big Y and a little y, that represents the same gene, but different traits. So for example, one might represent black hair, and little y might represent brown hair. You can inherit either the black hair gene or the brown hair gene. Again, they're independent of one another. They segregate, they split apart during meiosis, so you can only inherit one of those from each parent. Now, genes are pieces of DNA that store information on making a protein. Okay, so I'm using some of these terms interchangeably here. And then allele is a form of a gene that's represented by the letter. So you can have a gene that codes for hair color, but you might have a brown hair allele or a black hair allele. You might inherit one of those from the parents. So genes is just that piece of DNA. The allele is the specific form of the gene, whether it codes for one characteristic or another. Now, again, we inherit 2 alleles for each trait, one from mom and one from dad. The term we use to describe if we inherit the same allele is homozygous. The root homo means same. So if we inherit 2 of the same alleles, we call that individual homozygous for that particular allele. If we inherit 2 different alleles, that is known as heterozygous. Hetero meaning different. Okay? Now also keep in mind that the law of dominance plays a role here. Think about back to the yellow and green that we previously discussed. The yellow allele masks the green allele. If we inherited 1 yellow and 1 green, the pea plant would appear yellow, not some mixture of yellow and green. The law of dominance states that alleles that are dominant will mask those that are recessive. The term dominant means that it will mask the recessive. Now, in order to express a recessive trait, you must have 2 of those. You must inherit one of those from each of the 2 parents. So take a look at some of the examples here on the right. Big E, we're talking about attached or detached earlobes. Big E is homozygous dominant, meaning that individual has 2 dominant alleles. That person has detached earlobes. Big E, little e, heterozygous, means that that individual has detached earlobes as well, because the big E, which is representative of the allele for detached earlobes, is dominant to that of attached earlobes. The only way an individual can have attached earlobes is if they inherit 2 recessive alleles, or 2 small e's, one from mom and one from dad. A couple more terms we need to make sure we understand. The term genotype and phenotype. I will use these quite frequently in class. Genotype are the alleles that you have. So those are the genes, the alleles that you've inherited. You're either homozygous dominant, heterozygous, homozygous recessive, the terms that we just previously used. The phenotype is what you actually see. It's the trait that's expressed. So the phenotype of an individual is going to be what hair color they have, what eye color they have. Look at the picture down below. The genotypes are 3 genotypes that are listed there. Big B, Big B, Big B, little b, and little b, or homozygous dominant, heterozygous, and homozygous recessive. The phenotype is what you see. So in this instance, we have a purple flower, and a white flower. Okay? So make sure you understand those terms because we will use them quite a bit in this class. Now, with this information, we can actually use a system called Punnett Squares to predict the genotypes of the parents as well as predict the genotypes and phenotypes of the offspring. So when we do this, we basically separate the alleles using the law of segregation. Remember, independent assortment says there's basically a 50-50 chance of you inheriting one particular allele from a set from each parent. And so we use the laws of probability and statistics to do this. Okay? So follow along. It's pretty straightforward on how to do this, and you'll be just fine. All right, so what's really important with this, and this is really the first time we've done this in biology, I've mentioned this term before, which is RTFQ, which stands for read the freaking question. Make sure you read it, and seriously, highlight and underline the things that you need to know how to do here. Okay? So I'm going to highlight information that's given to me in the problem, and then I'm going to underline the stuff that we need to figure out. So in here, it says that tall pea plants are dominant to short plants. It is asking me to cross a homozygous tall pea plant with a short pea plant. The question is asking me to determine the genotype and phenotype of the offspring. All right, so let's work through this together. Step number 1 is to pick a letter to assign each allele. You're always going to be given information like this in the problem. So tall are dominant to short. Dominant alleles are always represented by a capital letter, and recessive alleles will always be represented by a lowercase letter. So tall pea plants, we'll just use capital T for that, are dominant to short plants, lowercase t. All right, we're off to a good start. The next thing it says is to cross a homozygous tall pea plant with a short pea plant. Homozygous means same. Tall. Tall is capital T. So to represent that, I'm going to use 2 big T's. That's homozygous for tall. Now, we're going to cross that with a short pea plant. In order to be short, remember short is recessive. In order to express a recessive allele, we have to have 2 of them. So your short pea plant will be 2 lowercase t's. It says determine the genotype and phenotype of the offspring. Now, this is where we build our Punnett square. Okay? So what we're going to do is we're going to make a square like this. So we're going to have 3 lines going down, and we're going to have 3 lines going across. We're going to take one of the parents, and we're going to place them at the top. Okay? Now remember, law of segregation says I can only inherit 1 allele from each parent, so we're going to split that. So we're going to put 1 T here and 1 T here. And then we're going to do the same thing with our short pea plant. So put a lowercase t here and a lowercase t there. All right? Now then what we're going to do is demonstrate fertilization, which means that sperm meets egg. So we're going to take each individual single allele and we're going to recombine them back together in the 4 squares down below. So in this top left square here, we're going to have a big T and a little t. And then in the next square to the right, we're going to have a big T and a little t. And you see how we're recombining them together. We're putting them back the way they were. So you just kind of take the squares, you kind of use it as a grid, and you put those 2 letters together. Okay, now it's asking us to determine genotype and phenotype of our offspring. So here's how we write that. We have genotype and phenotype. Now the thing to keep in mind is that when we do monohybrid crosses, there are 3 possible genotypes. I can either be big T, I can be big T, or little t, little t. Okay, those are the 3 possible genotypes. I can be homozygous dominant, heterozygous, or homozygous recessive. We'll take a look at that box there, and how many of those are homozygous dominant? Well, you would say that there are 0. There are 0 that are big T, big T. How many of them will be heterozygous, meaning that they have 1 of each allele? Well, that's 1, that's 1, that's 1, and that's 1. So then I would say that there are 4 that are big T, little t. And as you can already take a look and figure out, there are 0 that are little t, little t. Okay? Now this can also be represented by a ratio or a fraction or a percentage. So you could say 0 to 4 to 0. Okay? I could also say 0 out of 4, 4 out of 4, 0 out of 4. I could also say 0, 100, 0. Read what the question is asking for, because it may ask you for percentage. It may ask you for some of these things. So keep those things in the back of your mind. The phenotype is what we see. Well, there are only two choices for phenotype. I'm either going to be tall or short. So I take a look at that. How many of those are going to be tall? Remember that the tall allele dominant to the short allele. So all 4 of these are going to be tall. If you're heterozygous, you will be tall. And 0 will be short. Okay? So hopefully you have a little bit of idea of what that looks like as we go through and complete this problem. Let's do a few more. Let's follow the same process as last time. Tall pea plants are dominant to short. We're going to cross 2 heterozygous pea plants. And it's asking us to determine the genotype and phenotype of the offspring. So let's do the same thing we did with our last one. Let's, step 1, assign letters. Tall pea plants, again, we'll use a capital T. Whoa. We'll use a capital T. To short pea plants, lowercase t. It's asking us to cross 2 heterozygous pea plants. Remember, hetero is one of each. So this will be big T, little t. And we're going to cross that with big T, little t. Determine the genotype and phenotype of the offspring. So again, we're going to do the same rules as last time. Except we're going to take each of these and put them in the boxes. So big T, little t, big T, little t. Now again, we recombine them back together. If there's a big letter, always make sure you write it before your little letter. Okay, so we're going to determine the genotype and phenotype of our offspring again. So again, there are 3 possible combinations here. There's going to be homozygous dominant, heterozygous, and homozygous recessive. How many in the box here do you see are homozygous dominant? 1. One of those is homozygous dominant, so put a 1 there. How many are going to be heterozygous, meaning there's one of each allele? There's 1, 2, so we're going to put a 2 there. And how many are homozygous recessive, meaning they have 2 lowercase letters? There's just going to be 1. Okay? So now let's look at our phenotypes. Remember, there are 2 possible phenotypes. I can either be short or tall. How many of these are going to be tall? Well, that one will be tall, that one, and that one. Remember, if it has a tall allele, it will be expressed. So there's 3 of them. And short is then going to be just 1. This is the only one that's going to be short. Okay? So again, follow each step. Think about what the terms mean. Homozygous, heterozygous. Remember, genotypes, there are 3 possible genotypes. And there are 2 possible phenotypes. And before we start our third practice problem, just a little reminder that our word for the video is brown. Okay, the video word for this is brown, so make sure you include that on the front or back of your video sheet as we move forward. All right, so let's try one more practice problem. Brown eyes are dominant to green eyes. A man that is homozygous dominant for brown eyes has children with a green-eyed woman. Is it possible for them to have children with green eyes? So again, let's kind of follow the same procedures here, but read the question a little more carefully. So brown eyes are dominant to green eyes, so let's assign them a letter. Let's go with B. And green eyes are recessive, so we'll use a man that is homozygous dominant, so that's going to be 2 big B's, has children with a green-eyed woman. Remember, the only way to have green eyes is if we have 2 lowercase or recessive alleles. Is it possible for them to have children with green eyes? Now, some of you may be able to just look at this and tell. Let's go through a problem and let's build our Punnett square just to make sure. So we're going to take each of these and we're going to put them in here. Recombine them. All right. So is it possible for them to have children with green eyes? Well, if we take a look here, this child here will have will have brown eyes. This child here will have brown eyes. Again, the dominant masked the recessive. This child here, brown, and this child here, brown eyes. So all of them will have brown eyes. So is it possible for them to have children with green eyes? No. Okay. So again, I may ask you a couple different types of questions with this. You may have to actually work backwards as well. So just kind of keep these things in mind as we go through and practice some more of these Punnett square problems. So keep in mind that Mendel's research showed that traits are inherited separately from one another and from parents to offspring. And these alleles can be dominant or recessive, and that determines which traits are actually expressed. So remember, in an individual that is heterozygous, the dominant masks the recessive. Punnett squares are used to predict potential genetic combinations of offspring, just like we've done with the last 3 practice problems. A little bit longer video today, guys. Hopefully you got the gist of it. I won't scare you with some crazy opening like I did this time, but I don't know, something to keep you on your toes. Anyways, have a great day, guys. We'll talk to you later. Bye-bye.